All right, let's get started. So this lecture will be a statistics review. So many of this material in today's lecture, many of you might already have learned it during your college or any statistics courses, but it's just for a review from a computational biology perspective. And you can have access to the slides on the course website. And a few reminder is one thing is about the assignment. So the first assignment has already been posted at the website, which includes the assignment for both the Monday lecture as part A and today's lecture as part B. So there are two sections for this assignment. Also, so at the end of this lecture, we'll have a few minutes for you to work on the assignment, just in case you have any questions. So besides that, I'll just get started. So an outline for today's lecture. So we will briefly go over the concepts about random variable, probability distributions, central limit theorem. Central limit theorem, this part is directly related to this part of the assignment, the homework. Also, hypothesis testing, p-values, multiple testing correction, type 1, type 2 errors, and false discovery rate, or FDR. So many of these concepts or ideas will be highly useful or important in computational biology research. So I first saw this diagram to explain what data science means. So it's an interdisciplinary subject in the intersection between computer science, math and statistics, as well as a domain knowledge. So to study data science, you want to study the specific subject. You need to have the domain knowledge. So I always like to adjust this plot to be more specific to computational biology or biomedical data science. So of course, the domain knowledge in this subject of data science or biomedical data science is to study biology. So whereas the other two important foundations are computer science and math and statistics. So computer science, as many of you already have some preparations or foundations, and also as Nathan's lecture on Monday to go over the history of computational biology, we know that it's a combination between computer science in the development of computer science and biology. But the math and statistics part is sometimes ignored or omitted during this in this process. But in the research of computational biology, statistics and math is super important. And why is that? So statistics is the theoretical foundation of machine learning and data science. So although we are it's a computational biology class, is that we are not directly studying either data science or machine learning. As every one of us knows that the machine learning data science has been widely applied in the study of biology or biomedical sciences. And to study any subject, any technology, machine learning technology or data science subject, statistics is the theoretical basis. Also, statistics is the bridge between experiments and theories. So just like any scientific subject, the goal is to find the fundamental rule of the nature, right? So we use observations or we design experiments. We collect the experimental data, but the ultimate goal is to find the underlying rule or principle of this the science subject. Or eventually we want to develop or identify develop new theories. So how to connect the observation or observational data or experimental data into in a more theory form. Statistics is the important link. It is to make sense, statistics and make sense from the experimental data. Also, and the third point is statistics is the bridge between observation, which is the data, and the discoveries, which is science, the real science part. So when we talk about statistics, we often talk about a model. So what is a model? So model model can be different things in different sense. So in this context, a model, generally speaking, a model can be can used to describe the relationship between different quantities. So imagine when you are reading any scientific paper, when you're reading a plot, reading a figure. So all the information you want to get from a plot, from a figure, is all to see I mean, the goal of plotting a figure to present in the paper is to show the relations between different quantities. No matter what you see, it's a bar plot or a dot plot, scatter plot, or a more fancier like heat map or anything. So the goal of this presenting a plot are all about to reveal the relations between different quantities. And what a more theory form about the relation between different quantities is a model. So the model can be a quantitative model or a mathematical model. So the simplest form is a linear model. So meaning telling us different quantities will have a linear relationship. So with a multiplier or a one coefficient can connect the two quantities. So a statistical model is a type of model that to describe the relationship between random variables. So random variable, random is the key property of such type of variable. So when we observe a quantity in biology experiments, we measure say the weight and the size of something that you're measuring, or in genomics, we may observe the level of expression of some genes that can be measured by using some number, some variable. But as we know, if we repeat such experiments, if we measure the expression level or transcription level of a gene in different multiple times in replicates, the number may not be always the same. So repeat a sequencing experiments, the number of sequences or sequence reads at certain regions will definitely not be the exact same number. So this variable itself contains some randomness, meaning that when you observe it multiple times, we have different numbers. So that's a random variable. So statistics model is to measure the relationship or measure the feature between random variables. And there's a very famous saying that I think many of you have heard about this. It's called all models are wrong, but some are useful. So model is why we say that all models are wrong, because all models have their limitations. It's only useful when we use the model to describe the relationship between the variables or the quantities within the scope of our study of our experiments. So with the underlying this this whole scope, the model itself is not a scientific theory. The model itself is not a Newton's law or general relativity. It's not a basic scientific theory. So models can be wrong, but some are useful. So the models we use the models to describe the relations, which may give us some hints about the real underlying science behind the data. So I already mentioned the basic features about random variables. So the randomness is the most important characteristic about a random variable, and all the quantities obtained from experimental measurements can be considered as random variables. Because for any experiments, we cannot, there's no way that we can guarantee that every time we measure this, we get the same number, we get the same data. So they all contain some variabilities, and we can consider all of them as random variable. And that which is exactly, we cannot simply do any experiment just once, just one shot. It's not enough, right? We need to repeat them and get multiple times, and then use statistics or use any other approaches to analyze the data and to get the underlying truth from the observed random variables that we measure. And they are discrete versus continuous random variables, and we use probability distribution to describe the properties of random variables. So some examples about discrete versus continuous random variable, like if we count something, if something can be counted, they are usually discrete random variable, right? It's always integers, say, including in the genomics, in the sequencing, next generation sequencing data, the number of reads, essentially they are discrete random variables. The number of sequences is a count. So the count cannot be a continuous real number. It's always an integer. So one can consider it as a discrete variable. Whereas for continuous random variable, it's a real number, like continuous from mathematics. Say if we measured a weight, a height, a length, or something, it can usually be considered as a continuous random variables. And probability distribution, we all know that they are used to describe the property of random variables. So here are some useful discrete probability distributions that we see very frequently during our research or the studies. So, Bernoulli distribution is, well, yeah. So imagine that if we just toss a coin. When we toss a coin, what's the probability of observing on both sides? So that when we toss the coins case, so it's either the front face or the tail, but these two can be measured as either 1 or 0, and we can only have these two measurements. So that's why this is a discrete variable, and the probability of observing the 1 or 0 can be measured by a Bernoulli distribution, and it's generalized from tossing a coin, because for coin tossing, we usually think that the probability of observing either side should be 0.5, should be equal. But to generalize that, it may not be equal, right? So if it's not, say, a non-symmetric coin or anything, so on the one side it can be P, and the other side is Q. So P plus Q is 1. So when the probability of observing 1 is P, and the probability of observing the other side is Q. So 1 minus the probability of the other side is 1 minus Q. So binomial distribution is a more extension of Bernoulli distribution. And what is more useful in the, especially in the sequencing data analysis, is a negative binomial distribution. So which is commonly used to describe the number of read count or sequences in a certain region. If all of the sequence reads are distributed in certain regions in the genome, so for example, the gene expression. So if we measure the gene the expression level of some gene, of one gene, from an RNA-seq experiment, and repeat the experiment a few times. And then considering the expression, the total number of reads and are all the same, all comparable after all of the normalization processes, the read count we observed for that gene across different experiments should follow a negative binomial distribution. And the negative binomial distribution usually have two parameters. One is P and R, and they're used to measure the average or the mean or the variance in the probability. And also another very useful distribution is Poisson distribution. So Poisson distribution, we can consider it is a special case of negative binomial distribution when the we ignore the mathematical derivation part, but once the mean and variance is the same, then in that case, that is the negative binomial distribution can be generalized into a Poisson distribution. On the other hand, Poisson distribution is a very commonly occurred distribution in nature. So a lot of natural phenomena follow a probability for Poisson distribution. Okay, a few examples for continuous probability distribution. The easiest one is probably a uniform distribution. So it's across certain places, the probability of observing any value is equal. So it's a uniform distribution. And also a normal distribution or a Gaussian distribution is one of the most commonly used distribution in statistics because they have a very beautiful mathematical properties. And also we'll talk about the central limit theorem that also related to normal distribution. A normal distribution intuitively is easy to understand is that some things follow a normal distribution, meaning that we most likely can observe this around the mean value. And then the further from the mean value, the less likely we observe that value, right? Another useful distribution we observe frequently is exponential distribution. So exponential distribution is that something is the distribution of something is skewed to both ends. So the probability of observing very small value is very high, and the probability of observing very extremely high values can be very low. The exponential distributions also largely occurred in both the nature and the society, like in economics. All right. So the use of probability distributions in computational biology includes to establish or to set the null models for hypothesis testing. We'll go into details about both concepts about null hypothesis and hypothesis testing. Also, when we want to quantify or to characterize the signal from the data, or we want to also quantify or characterize the noise or background, and that's the only way we can detect either signals or signal enrichment or differential analysis. So when we see two quantities, if they are different, the question to ask is what do you mean by different? How different they are? Can we use some statistical or more solid way to describe the differences? So instead of directly saying that the two numbers are different, we can use statistical models or these distributions to build up models to further quantify these differences. Also, in the regression models or more advanced machine learning models, we always need to introduce the error term And the error models are usually described by certain probability distributions. All right, central limit theorem. So just briefly about the concept of central limit theorem is that regardless of the population distribution, so I didn't mention the population distribution versus sample distribution. So say population distribution is meaning that we get the measurement of all the elements or all the subjects in the whole population. So once we have the measurement of the entire population, we do not need to do any statistical estimation anymore, because the mean the population is everything we wanted to measure in the first place, right? Usually that's not feasible. That's not possible. So for example, if we want to get the average weight of all the human beings in the world, it's unfeasible to measure the entire population. Instead, we use a sample. So as long as the sampling process makes sense, right? We sample like 100 people, 1,000 people, or in the population context, we probably want to draw different samples or a group of people from different geographic places, or all different, consider them, and then use this as a sample to estimate the entire population. Yeah. So regardless of what is the population distribution, once we draw a sample, and then we can calculate the mean from that sample, right? The sample, we can draw the samples multiple times. We can get multiple samples, right? Central limit theorem tells us the sample mean will always follow a standard normal distribution if the samples are independent and equal size, right? So independent and equal size is some important conditions to apply on central limit theorem, right? So still, if we're using that same example of measuring the weight of all the human beings in the world, right? We draw the sample, one sample of 1,000 people, and then we independently draw another group of 1,000 people. So they should be independent, right? They should always be 1,000. And then we do this sampling process many times. And then every time we have 1,000 people, we calculate the mean weight of these 1,000 people, we get one number. And we draw another sample of 1,000 people, we can calculate their mean. And then we do this process many times. So all of the mean values will always follow a standard normal distribution. So that's what the central limit theorem is about. And also the important part is the central limit theorem is the theoretical foundation of many statistical tests. Most common statistical test is the t-test. Right? Once we, so central limit theorem guaranteed us that regardless of what are the original distribution, once we compare the two distributions, we can always use t-test. The t-test can always give us some information, always useful and commonly. All right. Next concept is about hypothesis testing. So I think one of the most important purpose of this class is to give us an idea about the concept of statistical testing and the statistical thinking, which means that statistical thinking can be different from general thinking, right? Generally, or intuitively, we think we want to ask some question, is that whether two quantities are different, whether they're different. So different two group of measurements, or in the genomics case, the expression of some gene in two different conditions, whether they are really different, right? Whether this differences in the condition or the effect of some treatment will affect the expression of certain gene, right? Whether we create we can result in differential expression. So general thinking. And then the question, and then the second, what are they different? And the second question would ask is the difference so-called statistically significant? And why we're asking this question? Because if we're only saying that they are different, we may not satisfy the reviewer. The reviewer will always ask you to provide some statistical significance. But I'm just joking. That's not the real. That's not the actual. The appropriate way of thinking this problem. So we want to do the more statistical way of thinking, which means the idea of null hypothesis versus alternative hypothesis. So instead of answering whether the two quantities are different or not, the statistical way of addressing that is that these differences, whether to compare with a null hypothesis, whether we can accept or reject the null hypothesis, assuming that the observed quantities, the data, are from some statistical distribution. Right, so that's the way of statistical thinking. So null hypothesis can be considered or can be understood as the negative control that we all know in biology experiment or any kind of experiments. So if we want to compare the effect of adding water to grow some plant, right? We always need to know to design. This experiment, we always need to keep a negative control group, right? So we have two identical pots and put the same amount of seeds in each pot, but we use different treatment, right? One with water and one without water. And then we can see in those pots, we have the sprout, and the control, we don't have sprout, right? We cannot say that if we without the control group, we do not know the effect of adding water. And we can only draw a conclusion if we have a control group. So null hypothesis is just like the negative control in our experimental design. So there are some conceptual argument or different ways of understanding what is null hypothesis. And some of them may not be accurate, or some of them may even be wrong. So in the statistical hypothesis testing context, null hypothesis, null hypothesis is a hypothesis, or is the belief beforehand, before we design an experiment. We need to know what is our null hypothesis, right? And the null hypothesis does not depend on a test procedure. So regardless of what treatment we have, the negative control is always there, right? So this way is saying, just take this example from different places on the internet, saying that the null hypothesis is a type of conjecture used in statistics that proposes that there is no difference between certain characteristics of a population or data generating process. And this statement is actually false, because the null hypothesis does not depend on any processes, any test process. Null hypothesis is there, regardless of what test we do, and regardless of what the results from what hypothesis testing we do. So null hypothesis is something we believe and cannot be, in fact, null hypothesis cannot be tested whether it's true or not. We can only from our using our data, our observer data, to accept or to reject a null hypothesis. So let's say there's another saying, which is hypothesis testing provides a methods to reject a null hypothesis within a certain confidence level. Right. So this is true. So null hypothesis cannot be proven. Right. We cannot prove whether a null hypothesis or is true or not. We can only make a conclusion from our observed data about whether we want to reject or cannot reject a null hypothesis. So statistics cannot tell us everything. Yeah. One main reason is that because the data is random. So different times we observe different type of data, we may draw different conclusions because we believe the data and the null hypothesis is our belief, right? It cannot be tested. Some other part that null hypothesis is misunderstood. Let's say this, on the left one, hypothesis that proposes that no statistical significance exists in a set of given observations that is used to assess the credibility of a hypothesis by using the sample data. Yeah, which is very confusing. But the point is that null hypothesis should come before any observing any data or doing any hypothesis testing. So null hypothesis is there regardless of whether we observe data or not, or whether we do a testing or not, right? On the left, a null hypothesis is a form of hypothesis that is deemed true until proven wrong based on experimental data. Generally, we usually think of a similar way of how to use a null hypothesis. But the confusing part here is that null hypothesis is there, regardless whether we cannot prove whether a null hypothesis is true or not, is right or wrong. It's just the only thing we can do is to compare the null hypothesis from our observed data. Right. So there are certain types of commonly used hypothesis tests, including students' t-test. Well, all the hypothesis tests include the two categories of hypothesis testing: is parametric test and a non-parametric test. So parametric test, meaning that in the statistical model or in the null hypothesis, there are certain parameters. So for t-test, which is a parametric test, and just to remind you to always keep in mind about the statistical thinking about null hypothesis. So the meaning of student t-test is to test whether the null hypothesis can be rejected. And the null hypothesis for t-test is that the sample means, the means of two samples, they are equal. So t-test, usually what we do is to compare the two group of numbers, and we use the t-test and generate a p-value. But this is just to remind us the statistical way of thinking. We always think that the meaning of t-test and the p-value will come out from the t-test. We'll discuss p-value later, meaning that we are accepting or rejecting the null hypothesis that the sample means between these two groups are equal or not, right? And Fisher's exact test is another useful hypothesis test, and the null hypothesis is the two groups are equally likely to observe a certain type of event or to possess certain feature. So Fisher exact test, basically to test whether these two groups, their chance of having some feature is equal or not. Or in other words, the separation of groups and separation with having or does not having certain feature is related or not. And Wilcoxon test, Wilcoxon rank sum test is a useful statistical test. It's a non-parametric test to test whether the two samples has the equal. The null hypothesis is that they have an equal chance of observing the one is bigger than the other or less than the other. Or in other words, it's a way. So when we do a t-test, it's a parametric way. It considers sample means. But the rank sum test means that whether we can so if there's one sample and we randomly draw the number from the one from this sample from these populations multiple times, and then we do not observe, we do not expect that the one sample is necessarily either greater than or smaller than the other sample. In that case, it's a null hypothesis case for Wilcoxon test. But if we draw the samples in a biased way, we can draw some samples that are greater than the other. And that is the case when the Wilcoxon test will tell us whether these two samples can be different. And another useful test is Kolmogorov-Smirnov or KS test. KS test for data visualization is easy. I mean, it's more straightforward to observe once we can calculate the cumulative distribution, which there are like two sigmoid or S-shape curves. And the goal of KS test is to test whether these two distributions are same or not. So in this case, the null hypothesis is that the two samples have the same cumulative distribution. And now is we cannot talk about p-value without having a clear concept about hypothesis testing. So having hypothesis testing, the test statistics and the p-value are the two usually most useful and important statistical quantities, especially the p-value. So the definition of, in this case, the definition of p-value in the hypothesis testing context is that the probability of observing the cases that are from the null hypothesis, right? If so usually if we want to observe the signal that are very different from the null hypothesis, we are going to reject the null hypothesis, right? So okay, so this is actually a small test, and I already gave you a hint about the answer. So which of the following statements about p-value is true? So based on your understanding from previous statistics classes and what I described just now. So A, p-values measure how big the difference is between the data sets compared. B, p-value is the probability of observing the data by random chance. And C, p-value is the least probability of observing the data under the assumption that the null hypothesis is true. So yeah, I can see your answers in the chat box. Yeah, feel free to. So there right now I received 4 answers, and yeah, you can feel free to type in your answer in the chat box. Yeah, I'm not creating a poll in this case. So yeah, so good that nobody chose A, and but probably B and C are equal. Yeah, probably there are more people chose B than people chose C. So the correct answer is C. Right? Why B is not correct? So that's the way we usually have the misunderstanding about p-value. So p-value is the probability of observing the data by random chance. So random chance is not necessarily true. What if the null hypothesis is not random? Say even from a t-test, right? If we compare the sample mean between the two samples, they're not random. The p-value does not have anything to do with so-called random. Instead, p-value, the correct answer is C. So p-value is the least probability of observing the data under the assumption that the null hypothesis is true. So in many cases, we just in general, we thought that the null hypothesis is some type of a random distribution. And in that case, we sometimes generalize, or it's actually a misconception. We consider the null hypothesis as a random chance. But in the more strict or more rigorous statistical thinking, B is incorrect. So p-value does not necessarily mean to observe data from random. P-value only means the chance, the probability, the least probability to observe the data under the null hypothesis assumption. So that's why when we observe data, when we see the data from the paper, from publication, once we try to interpret a p-value, or if we run some computational tools to analyze some genomics data, some tools will generate a p-value, right? And when we interpret the p-value, we should always ask a question that what is their null hypothesis? And it's not necessarily meaning that random is their null hypothesis. Or for the same reason, when we read different papers or different paper measuring or they are studying the same concept or trying to test the same hypothesis using different approaches, and they may report different p-values. So the key question is that so one study reported a p-value of 0.01, and another study report a p-value of, say, 10 to the negative 3, 0.001. Does that mean that the one with 0.001 is more significant than another study that only reports 0.01? Right? The answer is no, not necessarily, because we don't know their respective null hypothesis. Right? The p-value only makes sense under that correct null hypothesis. So generally speaking, when we see different p-values on different places, they are not comparable. The p-values are not comparable. They're only comparable when they are under the same statistical test using the same null hypothesis. So that's the important part here. Okay. Yeah. So remember the answer to this question, as you might see this again sometime. All right. Any questions so far? Okay. Let's move on. Okay, so a few years ago, the American Statistical Association published a statement to the public, specifically to raise the public's awareness about the misuse of p-values in a lot of different scientific publications in the different places. So yeah, we'll just briefly review this statement about statistical significance and p-values again, because especially as a computational biologist, we should be rigorous and clear about the concept of p-values when either using computational tools or developing our computational tools. So number one, p-values can indicate how incompatible the data are with a specified statistical model. Two, p-values do not measure the probability that the studied hypothesis is true or the probability that the data were produced by random chance alone. Number three, scientific conclusions and business or policy decisions should not be based on only whether a p-value passes a specific threshold. Right? This is very common that we get the experimental data and do some tests, and when we see the p-value is 0.06, oh, too bad. Or if it's 0.05, okay, then it's publishable. It's very common in different places, but p-value simply passes a 0.05 doesn't necessarily mean anything, right? So that's raised by the professional statistical associations that we cannot simply base on those. And four, proper inference requires full reporting and transparency. In this p-value context, it means that it should clearly state that what test is used to generate the p-value and what is the null hypothesis if it's not clear. So that's why when we report our data, instead of simply saying the p-value is less than 0.05, we we clearly specify what test was used. And if necessary, if needed, also report what is the null hypothesis. And number five, a p-value or statistical significance does not measure the size of an effect or the importance of a result. So that's that's a concept of two different concepts in statistical test. So one is effect size, and the other is the p-value. So p-value only tells whether you want to reject the null hypothesis or not. But how big, or in other words, p-value tells whether the differences is statistically significant, but it does not tell you how big the differences are. It might be very different, but they are not statistically significant. On the other word, it might not be that different, but it still can be statistically significant, right? So we can, I'll give one example here, use the simplest case, coin tossing again, right? So if we toss a coin, say 4 times, although we know that the chance of observing a front or tail is equal to 0.5, but we observe like 3 tail and one front has a very high chance, right? But the difference is actually very big, right? If we only toss the coin 4 times. Instead, if we toss the coin 1,000 times, the chance of observing, say, 75, that's 750 one side and 250 at the other side. These comparison, the effect size are the same, like 3 compared to 3 face versus one tail. If we toss the coin 4 times, but in the 1,000 times, although under the same effect size, the statistical significance will be very, very different, right? Yeah, got that mean. Okay, so that means the difference, there are difference between the statistical significance and the effect size. Yeah. And number 6, by itself, a p-value does not provide a good measure of evidence regarding a model or hypothesis. So again, so this is the very beginning of what the means of a model, what it means of a hypothesis. The p-value itself only tell us whether we want the observed data we can use to reject or accept null hypothesis. But whether the model itself is true or not, or whether the hypothesis itself is true or not, cannot be told by the p-value alone. Okay, so regardless, well, if we have a clear meaning of what a hypothesis testing and null hypothesis p-value means, so they are very useful approaches when dealing with a lot of data sets. Is the multiple testing correction, right? Especially in high throughput or genomics experiments. So for example, once we, I mean, every time we do a sample and we can do a statistical test, we can observe a p-value. So multiple testing correction simply means that if we do this whole procedure many times, so even under the same null hypothesis, there's a high chance that we will observe will make the observation that is sometimes close to the null hypothesis. So in this case, we have to do the testing correction. So meaning that if we do the experiments many times, we have many p-values. The p-values, the p-value of certain number may not be as informative as we only have this one p-value. Or in other words, it may not be, it no longer be so significant compared to we only do the experiment once. So for example, in RNA-seq experiments, we can identify hundreds of differentially expressed genes from 20,000 genes in the, say, human or mouse genome. For ChIP-seq experiments, we can observe up to maybe over 10,000, say, peaks or transcription factor binding sites from the genome. So every differentially expressed genes or every peak from ChIP-seq there will be a p-value. But is that p-value means it is? Not necessarily, right? Because say if we observe a p-value of 0.01, 0.05, right? So every time we observe 0.05, meaning that there's a 5% chance this measurement is from a null hypothesis. But if we do this experiment 100 times, 1,000 times, the actual chance of observing the data from the null hypothesis will significantly increase. And that is why we need to do a multiple testing correction. So there are different ways of doing this multiple testing correction. So I will only talk about two ways. So one is Bonferroni correction. So Bonferroni correction, it controls the family-wise error rate. So it considers everything together. And it's a very strict way of doing the testing. It does not consider the different p-values across different observed results. So they to do the multiple testing correction or adjustment is to adjust the p-value by simply times the p-value by the number of tests we made. So n times p. So which can significantly increase the value of p-value. So it's very strict. See someone raise hand. Go ahead.
Why is the adjusted p-value n times p? Would that not increase the p-value that you're looking
Yes,
for?
it is. It does increase the p-value. So why adjust the p-values?
Oh,
I'll
it's
repeat that.
adjusting the p-value, not the significance threshold. Sorry. That's
Oh yeah.
okay, sorry.
So under this, when we do the model, so the goal is to give the p-value a statistical cutoff, right? The p-value cutoff. So once we have the observation, we do, I mean, the entire observed data is from many tests, right? Say we have 100 test. Every time it's under the same comparable null hypothesis, we generate a p-value, right? Remember that if we still use the original p-value of 0.05, which means that there every time there is a 5% chance that this is actually from null hypothesis. This is not the signal. This is from null hypothesis. But imagine 5% is 1 every 20 times, right? But if we do the test and I have the p-value of 0.05 20 times or 100 times, there is a high chance that some of them may be not true, or maybe from the null hypothesis, to be more strict. So multiple testing correction is just a way to adjust the p-value itself, so that after this adjustment, we can still use the original cutoff to give to select the significant from the non-significant. Yeah, did that answer your question?
Yes, thank you.
Okay. Yeah. So again, Bonferroni correction is a very strict way. So regardless of what the original p-value is, we just multiply this by n, by the number of tests. So for example, if we have 100 tests, there, original PO, the original number of 0.05 will definitely no longer be significant. So we need to have, originally we need to have a 0.0005 to be able to be significant as 0.05 after times 100, right? Another more commonly used way of doing multiple hypothesis testing is the Benjamini-Hochberg correction, or BH correction. So usually we have n different observations with different p-values, right? This is a way of considering the difference level of p-value itself. So the procedure is like we rank n. So from the smallest to the to the biggest, right? The rank on top is those that have the smallest p-value. And the way to adjust the p-value is that to times n and divided by the rank. So the highest one, the one that have the most significant p-value, will be the similar as Bonferroni correction, right? With just times n. But then as the p-value becomes larger, so there's another way to adjust that p-value. So so this is just the general idea, and the detailed calculation might be slightly different than this, because before and after this adjustment, they will also need to guarantee that the rank will not change. So the adjusted p-value and the original p-value will still have the similar rank. So so that's the way of what the more common way of doing the multiple testing correction, or Bonferroni-Hochberg correction. And this BH correction is used in many of the computational biology tools that we use, say DESeq2 for differential gene expression analysis, and say MAX2 for ChIP-seq analysis, and a lot of these high throughput analysis. And this is moderate. It's not as strict as the Bonferroni way, and it's actually a very sophisticated statistics theory behind this, and the procedure is very neat. Okay, we'll briefly talk about the concept. The concept about confusion matrix and the prediction and the truth. So that's related to the concept about type 1, type 2 errors and the false discovery rate. So so once we decide, so there are one way of statistical thinking is that we tend to believe there is an underlying truth. So also in this lecture, I'm not going to touch the difference concept about the frequentist statistics versus Bayesian statistics, but most of these is under the concept of frequentist statistics, in which we believe the nature has some underlying truth. And our way is to study. We want to use all of this experiment or test to find the underlying truth. So we know, so there is underlying truth of true, let's say, yeah, true or false, right? And then if we don't know the underlying truth, we design some test or some way of make the prediction. The prediction can tell us whether it's positive or negative, right? So then there are 4 different classes. True positive, meaning that underlying is true. And then we say it's positive. So that's true positive. That's the best. That's what we want. So is the true negative. The truth is that it's negative. And we also predicted it's negative. So it's called true negative and true positive. And then in the meantime, there are 2 types of errors. So one is that the underlying truth is negative, but we say it's positive. And so that's type 1 error. Type 2 error is that although the underlying truth is that they are positive, but we did not predict them as negative. So there are tons of examples from the well, I say easiest example is the disease diagnosis, right? So whether a person has the disease or not have the disease versus some diagnosis test, whether we can correctly predict disease or not versus disease or not. So type 1 error means that the person does not have this disease, but in our diagnosis test, we say they have the disease. And type 2 error means that the person does have such disease, but this diagnosis is failed to detect this, right? These are the the concept, these 2 type of errors. So extended from this confusion matrix, there are a lot of different different ways of measure a performance of a test, right? So the ultimate goal is so that our prediction can better reflect the underlying truth, so that we want our ideal case, the perfect case, is that the true positive 100% and true negative also 100%, and 0 type 1 error versus and 0 type 2 error. But that's the ideal world. That's the ideal case. In most cases, it cannot be this perfect. So then there are different ways of measure. So you can keep this whole summary as for reference, because some of the measurement might be useful in the especially to measure the performance, to quantify the performance of some machine learning approaches, like measure the accuracy. And there's accuracy, there's sensitivity or recall, and there's specificity. So sensitivity and specificity, and also I think there's important and also precision. And also there is an important concept about false discovery rate. Yeah, false discovery rate. The false again, so false discovery rate is the false positive one here, the false positive one, and divided by test positive, divided by everything that we call the test called it positive. So among all the ones that we a test reported, what the percentage of them or how many of them are false positive or false. So that's a false discovery rate. So under certain conditions or certain statistical concept, the false discovery rate here is is equivalent to the Benjamini-Hochberg corrected p-value or adjusted p-value. So sometimes in some context, these 2 concepts are used interchangeable. let's say, for example, in the DSeq2 model. So they use adjusted p-value, but then they use this cutoff as saying that this is the FDR, the false discovery rate. So this can be, in practice, this can be used interchangeably, but this is under some certain statistical model. All right, so I'll give one example. Not sure whether you get a chance to read this before the class, but this is a very interesting paper published a few years ago in Genome Biology by two of my friends, Jessica Lee and Wei Li. They collaborate. They found a very interesting observation. So the title is exaggerated false positives by popular differential expression methods when analyzing human population samples. So the interesting thing that they found is that the number of identified differentially expressed genes using the commonly used differential expression analysis tool like DSeq2 or HR, they are hugely, they are huge. But even after, even if they just randomly permuted the samples, say treatment versus control, random, they found that in this randomly permuted data, where we already destroyed the relations of different conditions, different disease or treatment versus non-treatment diseases, the conditions, where we should not expect to observe any differentially expressed gene again, anymore. But in many of these data sets, they still observe a huge number of differentially expressed genes. Yeah, which is worrisome. So, and then they found that the reason for that is that we need to go back and take a look at what is the assumption in these commonly used computational tools for differential analysis, including both DSeq2 and HR. Both methods use a negative binomial distribution as their underlying or so-called null hypothesis to do the test. So again, to reveal that negative binomial distribution measures in one certain condition across different samples, the expression level of one gene follow a negative binomial distribution. And then the null hypothesis is that the two genes in the two conditions, they are not differentially expressed, meaning that the average, the mean value in the negative binomial distribution, which measures the expected differential expression level in condition 1 and condition 2, they are the same. There are no difference, right? And these null hypotheses that used in DSeq2 and HR, they are only appropriate only if the negative binomial assumption is reasonable. Which in this case, which is true when both methods were developed, they are using like mouse data or cell line data, which in those cases, the replicates in certain conditions are very similar. And in those cases, the negative binomial assumption can be assumed, can be true. But in a large population or patient sample, the gene expression across different individuals are huge. And they use a QQ plot to compare the actual distribution of some gene expression with the theoretical distribution of negative binomial, and they found that the observed data, they already deviate from negative binomial distribution. So that negative binomial distribution assumption can no longer be held. So which is why the commonly used DSeq2 or HR can exaggerate the false discoveries and can observe a lot of differentially expressed genes, although in fact they should not be reported differentially expressed. So this paper, they actually proposed an alternative way, which is actually a much simpler approach. They just use Wilcoxon test. Remember that, or Wilcoxon test simply use the rank of the conditions and saying that whether these null hypotheses are the null hypothesis that the rank of one greater than the other or the smaller than the other is equal. So that's the null hypothesis, which is a non-parametric hypothesis test, and it's much simpler than the complicated parametric test. Yeah, and this Wilcoxon test does not have the negative binomial distribution assumption. So then they generated this using both the real data and the simulated data to prove that these approaches are simply using Wilcoxon test can outperform this more fancier tools like DeepSeek or Agile, right? And usually, but this case, they usually work when the sample size is large and the population data and the sample size is large. And they gave a practical suggestion that when the sample size is greater than 8, we can use Wilcoxon test. Okay, another interesting example, also included in the pre-class reading, is about Monty Hall problem. So I assume that many of you have heard of the Monty Hall problem, right? So I think the question that the problem is that there are 3 doors, right? It's a game, right? And one of the 3 doors, after one of the 3 doors, there's a car, and the other 2 doors behind the other 2 doors there's a sheep, so or goat, I don't know. And we assume that the player wanted to get the car, which is much more worthy than the sheep, right? So they have so at the beginning, the player opened 1 door, and the judge opened 1 door and find there is a sheep. So at this moment, they ask the player whether you want to change your decision. So they remove 1. So first, at the beginning, the player pick 1, right? And the judge opened 1 of the other 2 and found it's a sheep, and then ask the player whether you want to change your decision or not, right? So the one interesting part is about the Monty Hall problem is that the procedure actually is important. The procedure of whether they change or whether they make the decision of change before or after they open up the door. They open up the door. So yeah, you can read through that interesting paper from Nature Biotechnology. But the idea is that when we do all of the statistical analysis, the order of action actually matters, right? So this is using real genomics data as an example. So when we do the say differential expression analysis, we usually calculate the p-value, and then we have another p-value thresholding, right? And then after this one, we use the FDR or BH correction to select the top features, and this process is correct. But the incorrect way that some people might do is that, so after that, after the first round of p-value, and we select some top features, and then in this selected top features and use use a second p-value thresholding, and then reported this one as a p-value, and then this is no longer true, because once you change the order of action, in this particular case, the FDR is no longer have the same meaning as the original FDR, because the entire observation, you already screened the data, so you look at part of the data, and the entire story is no longer. The assumption of Benjamini-Hodgkin correction is no longer hold. Right. Okay, And at the end of this lecture, we will just quickly go through 10 simple rules of effective statistical practice. So there is a series of editorials published in PLOS Computational Biology over the years. They cut a series of 10 simple rules. So 10 simple rules for many different things. So you can, a lot of these articles are very interesting to read. So this is one example about statistical practice. So with or without very strict or rigorous statistical training or statistical class, we should always follow this statistical practice when doing our scientific research. So including number one, statistical methods should enable data to answer scientific questions. So yeah, so this concept that statistics is not simply a way asked by the reviewers to add a p-value, right? And signals always come with noise. So that's also kind of a belief we should keep in mind before we look at any data or design an experiment. Number three, plan ahead, really ahead, meaning that a lot of things should occur when we do the study design, when we do study with the experimental design. As a computational biologist, sometimes we start with the experimental data generated by our collaborators or publicly available data. So in this case, sometimes once we found that, well, they really should have designed the experiment in a different way. So then we should just tell them that what is the better way of design the experiment to generate the data that easier for or better for to making the conclusion. And we should always worry about data quality. So when analyzing any data, assess the data quality, do the QC as the very first step. Number five, statistical analysis is more than just a set of computations, right? So statistical thinking, like what we repeated a few times, is more important than simply following some procedure to do the computation. Yeah, number six, keep it simple. So do not think that statistics is something very complicated. And once we make clear about the concept, simple approach is always better than complex approach. Number seven, provide assessment of variability. It's also another point of statistical thinking. All the numbers come with variables, variances, and so consider the variability from the data. And number eight, is helpful, always go back and check your assumptions, not only when calculating a p-value, but also when making any conclusions, whether the conclusion is only true under some underlying assumption that we sometimes may or may not notice. Number nine is when possible to replicate, is to emphasize the importance of sample size. And number 10, make your analysis reproducible. Oh, well, that's everything that's important for now. I like to use this cartoon to end many of my lectures because not only because what I give you in any type of lecture is just a oversimplified version of instruction, but compared to the small details. But in this context, in this lecture in particular, which I just want to use this to emphasize that when we do all of the data analysis or design or do the experiments and do data analysis, to record the procedure details is very important, and that is also important to for reproducibility, right? Ideally, I always said that the expectation is that the readers or reviewers can repeat and reproduce all the results that you have. So that should be the level of details in the paper that we write. All right, I'll just end here. About assignment 1, well, I'll especially talk about section B. Please remember to record all the code you use and the results, and include sufficient amount of comments and explanations in the code. Or after getting the results, you can add the comments that you want to add. And you can submit this in any format. but R Markdown is preferred, but you feel free to submit it in a different format. And you can just email me the assignment when you're done. All right, I'll just end here, and we have a couple of minutes left. So feel free to stay in the Zoom if you have any questions or you want to work through these the homework in the next few minutes. Feel free to do that, and I will stop.